We are so excited. This is such a great day because we've never done anything like this before. This is the first POLG Summit, and we use the word summit very strategically. We've got a mountain to climb, and we're doing it together. I'd like to introduce Frederick's father here. Robert, if you'd care to say a word or two.
Yes, greetings everyone. And we're talking about humility, the humility of seeing you all around this screen here. This is all about solutions. I'm here to listen today because the work that you, Brian, are doing, Paola, Luca, and all of you is what we're all about. Our mission is to find a cure, treatments, and to be able to get rid of this disease. As you always say, Brian, our best success is putting ourselves out of business, and that's what we're counting on doing thanks to all of you. So that's my 15 seconds, Brian, that I told you I would give, because this is all about putting ourselves out of business thanks to your work. Thank you for your work, and thank you for giving us hope.
Bravo. Thank you so much, Robert and Julie. I'm going to share screen real quickly and just show you our agenda, but I wanted to also make sure I had a chance to acknowledge. I'm hoping you can see my screen. And if that's showing well, then I'd like to make sure you know who Luca Bolliger, our Chief Scientific Officer is, Paola Casanova, myself, and our dear hero, Frederick. And I would like to take just a brief moment, just a moment of silence to honor our inspiration and my co-pilot, Frederick.
Are you going to sit in here and play this? Have you got your light on?
Right, thank you all. Here is our agenda for today. If you can see this, I would appreciate everyone could try to stay on mute. I think I am able to mute everyone if I need to from the host position, but I don't really want to do that because I won't be able to keep track of who's who. But if you have questions, please type them in the chat room, the chat section, and Paola and Connor will help me try to sort them through.
And by the way, Pete Alonso doesn't even qualify.
And I hope that's okay. Sorry, I just had to mute someone. I wondered if we had any scientific advisor board members, board of director members on the call they also wanted to acknowledge, because we have such a superstar group of people that are the backbone to our foundation. I'm flipping through this. Oh my gosh, I see so many rock stars in the POLG world on this call. All right, well, I will pick on names of people that I wanted to point out, but here's our agenda. Everyone's going to have 10 minutes to speak, with the exception of the Pioneer team, with Omar Hikmat, Amel Karaa, and Phil Yeske. They'll go 15 minutes. Luca will talk about our toolbox and our funded research projects. Stephanie Harry and Kira Mund from MitoAction will talk about two subjects of great importance. And then Drs. Amy Goldstein and Mary Kay Koenig will present the POLG Crisis Response Team. And Robert Pickathley from UCL will speak about the LifeArc UK gene therapy work. And then we have industry partners who are here also to potentially share their updates or just participate and listen. And then if there are those that might be interested, there's a bonus 30 minutes at the end of this. If anyone wants to stay on and have a chance to debrief and chat with Stephanie Harry of MitoAction, please feel free to do so. So to start us off with, I wanted to take us back to a little history. And Bob, I know you're on the call. I don't know if you want to unmute yourself and say hello, but I wanted to acknowledge that the foundational work of which Bob Navio and the team with Richard Haas in 1999 were the first to make this association that a mitochondrial DNA polymerase ties into what is Alpers and in the spectrum of POLG disease syndrome. And the spectrum is, as we all know, very complicated. But Bob, I wanted to make sure that you were recognized today at this first POLG Summit, because I don't think we would exist if this really groundbreaking work didn't start off 26 years ago or got published 26 years ago. So Bob, I don't know if you want to say hello or anything like that.
Yeah, thank you very much, Brian. I'm just excited to be here to hear about all the progress that's being done. So I'm looking forward to the day.
Wonderful. Thank you for joining. And then I have to also recognize Bill Copeland. And Bill, I believe you're on the call too. Some of the work that you've been extending beyond is really helping us understand all the mutations, of which POLG has such a complex story. But this database, this POLG mutation database out of the NIH, is run by Bill Copeland, and have done some really scintillating work of helping us really understand what is it about the functions of POLG and where the mutations happen to lie. And Bill, I don't know if you want to say hello to everyone. Please, I welcome it if you would.
Sure. Thank you, Brian. Unexpectedly, but this is great. I just want to give a cheer to everybody and for you guys, you and Julie, and for putting this together. This is a phenomenal summit. I'm looking forward to it. But thank you.
Wonderful. All right. Well, we're not done yet. We're going to get rolling here. If I could invite the pioneer team, Amel, Phil, and if Omar's on the call, I'll take it to you all. Please tell us about the pioneer study.
All right, Brian, thanks very much. Hi, everybody. Phil Yeske from the United Mitochondrial Disease Foundation. I'm going to kick things off for the team, but you will be hearing from Doctors Karaa and obviously also from Omar as well. Let me see if I can get our slide shared here, Brian. Look like that's working okay?
Yes.
Great. So again, real pleasure to be here, and certainly everything we're doing is in honor of Frederick and all the other POLG patients that have gone before him. But also, we hope to be an inspiration to all those currently diagnosed with POLG disease and try to make a difference in your lives and those in the future as well. I'm just going to start out with a brief introduction of UMDF, since this is a global meeting and many of you may not be familiar with our organization. We're actually the oldest and largest patient advocacy group that's really an umbrella organization covering all forms of mitochondrial disease, but we have been serving the mitochondrial disease research community since 1996. We have a very broad mission. We're very proud of the fact, particularly for me from the research side, that we've funded directly over $17 million worth of research into labs around the world, not just in the United States. More recently, we've initiated no-cost genetic testing programs to try and close the gap. This is currently for those residing in the United States who may not have access to genetic testing. So if you're US-based, don't have a diagnosis yet of mitochondrial disease, a genetic diagnosis, please check out the link shown there to see if perhaps you qualify for that. But support, education, awareness, all very important parts of our mission as well. We're really thrilled to partner with all the families across all forms of mitochondrial disease, including POLG disease, to help support you on this journey with mitochondrial disease. And really, it's all about leveraging, whether it's POLG Foundation or UMDF. We know we only have so many resources available, but it's the ability to make our investments in the research ecosystem and in turn have those investigators go out and raise even more money in the form of grants to conduct their research. So we can see our 17 million has turned into over 160 million of additional follow-on research. So we're really proud of that fact. I share all of this because it really establishes why UMDF and why therapeutic development is so important to UMDF. We've really kind of lumped all of our therapeutic development efforts under an initiative called Treat Mito. So this is where we will try to bring all the clinical investigators and other stakeholders, patients, patient advocacy groups, industry, together to try in a more coordinated way advance therapeutic development in mitochondrial disease. You see the link there if you want to check out more about that. But really the most important project and how this ties together with Pioneer is our worldwide patient-populated registry called MitoShare. This is the only worldwide patient-populated registry that exists for mitochondrial disease. And we're really proud of the fact that over the last 3 years, we've grown this research database. So every caregiver, every patient that joins signs an informed consent to participate in research so that we can share their de-identified data in a way that advances mitochondrial disease research. So in this time, in approximately 3 years, we've grown MitoShare to over 3,100 participants. We have 50 plus countries represented inside of the registry, and hundreds of mitochondrial disease types are represented. I can't emphasize enough that this is a registry for all. You do not have to have a confirmed diagnosis of mitochondrial disease to join. In fact, if you're early in the journey and you just want to have a community where you can join and be a part of research, please do. And we will do everything we can to help you on your journey to eventually either having a confirmed diagnosis of mitochondrial disease or perhaps some other genetic disorder. But because we've built this research database, this really enables us to be able to facilitate clinical research out of this database, specifically natural history studies and clinical research studies. And through that, we've done several studies already. The first was the MitoArch study in collaboration with Tseng, developing a therapy. We're currently in a project together with Abliva, now part of Farming, called the Merlin study. So these are studies where we're collecting data that can then be used either in a regulatory context or just to advance mitochondrial disease research in general. But importantly for today's meeting, we have a really important collaboration in place with the POLG Foundation, where UMDF and MitoShare will serve as the stewards of the natural history data collection for the Pioneer study that you're going to hear more about shortly. So really, it's this opportunity, together with Pretzel Therapeutics, who is also providing some funding to be able to collect additional layer of data. And we're really thankful to the UMDF donors that have supported this project as well. This is where we need your help for the patients that are on the call. We need more POLG patients. Yes, the clinicians will bring patients to the registry, but the more patients that we engage in clinical research, the better prepared we'll be. So from a clinical trial readiness point of view, to conduct clinical trials, investigational therapy clinical trials on POLG patients with potential therapies there. So please, if you haven't already checked out MitoShare, the link is shown below, and we really hope as many of you as possible will get involved. And I think with that, I'm going to turn it over to Amel next.
Thank you, Phil. I am going to also share some slides. Hello, everyone, wherever you are. Good morning, good evening, and good afternoon. I am here with Omar. Omar, are you able to log in? Because he was having some trouble earlier.
I've texted him some more link information, so hopefully he'll be able to join shortly. But why don't you go ahead and proceed?
So I am here to start us off by introducing the Pioneer study. This is a jointed effort by the US and Europe, led by Omar Hikmat, my colleague from the University of Bergen in Norway, and myself in Mass General Hospital. And this study has been in the making for almost 3 years now. The very first step was the POLG Foundation put many of us experts in one room back at when the Euromit meeting was happening in Bologna, and they told us that we needed to do something about POLG disease, and we brainstormed for many hours. We all agreed that a natural history study was the first step to build data to better understand the disease, how it progresses, what the symptoms that are most meaningful to the patients are, and how we can fix them with new drugs. we agreed upon a list of data that needed to be collected through this natural history study. And over the last 2 years, we've been working towards this goal, and I'm happy to report that we are at the final stages of getting this started in the next few months. So the the natural history study aims to determine the POLG disease using prospective clinical laboratory and patient-reported data. It also aims to validate reliable outcome measures to evaluate the effectiveness and the safety of novel therapies, all of this spanning to several continents. So from on our side in the US, as you heard from Phil, we are going to be using the MitoShare registry, and we invite everybody who has a POLG disease or any caregiver of a POLG patient to enroll in MitoShare, because we really want this to be the POLG registry where all patients around the world can come in, register, tell us about their problems, and answer some questionnaires. And from all those patients, we are going to select patients that we will be enrolling in the PIONEER study. And for the PIONEER study, we are looking for patients with genetically confirmed POLG disease, aged from birth to 75 years old. And those patients are going to be evaluated through our selected sites by using patient-reported outcomes. So we will have a patient portal into MitoShare where we are going to ask patients to enter their own symptoms, what they are feeling, what they are living with every day. We are going to ask them to provide us with their genetic information so we can make sure we understand the genetic underpinning of their disease. We are going to collect some samples from the patient. We are going to ask them to fill out surveys about their quality of life, about their symptoms, about how they are feeling. And we are also going to ask their caregivers to also report on their lived experiences taking care of someone with POLG disease. In parallel to that, we are going to ask clinicians to also evaluate these patients in the clinical setting by doing examinations, reporting back on what they are seeing as clinician in the patient, how the patients are performing in terms of walking and their balance, and any problem that they might observe. And those data is going to be entered by the clinician into MitoShare as well. And both the patient and the clinician information is going to be gathered together under the same patient's portal. We have 7 sites in the United States who will be who will be doing this PIONEER study: Akron Hospital, the Cleveland Clinic, Columbia University, Baylor Medical Center, UT Health System, and Children's Colorado Hospital. And we are planning for this study to start for 3 years, and we are hoping to continue this as long as we can. Patients will be asked to come regularly for clinic visit at least once a year in person, and they will also have virtual visits and telephone encounters. And with that, I'll turn it to Omar. Omar, are you here?
Yes. Do you hear
Okay.
me? Is everything okay?
Yes, we
Yes.
can hear you.
Good afternoon, good evening, everybody. I'm Omar Hikmat. I'm pediatric neurologist working in Bergen, Norway, and associate professor at Bergen University. And for more than 15 years, my clinical and research work has focused on POLG disease. And during this time, I've been deeply involved in caring for affected children and family, and doing some study in natural history and spectrum and clinical spectrum of this disease. I'm truly excited to be part of this prospective natural history study. This initiative represents an important step forward for our field. As Amel mentioned, this is a global effort, and it is absolutely essential given how rare this condition is. Patients will be recruited from several centers across the world, and every single patient involved in this study will follow the same predefined study procedures. This include, as Amel said, clinical assessment, functional evaluation, and also systemic collection of patient-reported data. Having this harmonized data collected across multiple international centers will allow us to compare patients meaningfully, identify pattern of disease progression, and better understanding of factors which influence the prognosis. This is also help us to define reliable clinical endpoint, which is absolutely critically for the design of future clinical trials. Without high quality natural history data, it's extremely difficult to evaluate whether a new therapy is truly effective. Another key strength of this study is the inclusion of patient-reported outcomes. Listening to the patients and family is essential because clinical tests alone do not capture the full impact of the disease on daily life. By integrating patients and caregiver perspectives, we will gain a much more complete understanding of how POLG disease affect the quality of life, physical function, and long-term well-being. Importantly, this is not just about collecting data, it is building a collaborative international community. Amel, could you move to the next slide?
Yep.
In my network, we will recruit patients from 18 centers across 9 countries, and we hope that even more centers will join this effort. The stronger and the broader this network becomes, the more powerful the data will be, and the faster we will move toward meaningful advances for patients. So the goal of this project, as Emma said, is significantly improve our understanding of the natural history of POLG disease and create a strong scientific foundation for future therapeutic development. We will also share the same ambition to move beyond supportive care, which we have today, toward more disease-modifying treatment. This study will play a critical role in making this vision a reality. So I would like to thank the POLG Foundation for funding this huge project and also thank the investigators, clinicians, and patients and family for their contribution. I'm very proud to be a part of this global collaboration, and I'm confident that together we may make a real difference for individuals affected by POLG disease. Thank you.
Thank you, Omar, Amel, and Phil. I would be remiss to also introduce you to the POLG clinical operations team, and that would include Mindy Leffler, Dr. Harsha Rajasimha, and Adarsh. I don't know if they're all in, but if you could at least raise your hands and maybe just say hi real quickly.
Hi, everyone. Really inspiring effort and initiative, Dr. Tseng and the POLG Foundation. Really excited to see so many experts in POLG disease all together here. So very encouraged to see the future of finding a cure for this deadly and horrible disease. So happy to support the natural history study, and others might not be able to join. It's almost 2 a.m. in IST, but I was able to join from the Eastern time zone.
Thank you. Thank you. Mindy, would you like to say hello and a bit about your role with the Pioneer Study, if able? I know Mindy is traveling, so maybe we'll hold off on that. So moving right along, you heard a lot from Phil on the UMDF. I also want to recognize some amazing other partnering patient groups that are on the call right now. I'm seeing Pedro Mendez from MitoCon Brazil. Greetings, Pedro. I see Kate Murray from Mito Canada. I see Doug Lingard and Margie and Sean Murray from Mito Foundation Australia down under. Delightful to know that you're here with us in spirit and virtually. Andy, Bethany, Kara, Margaret, and Kristen Clifford from the UMDF. Amazing occasion that we could all be together virtually. I wish we were all in person, but I just want everyone in the world, in the community, to know this is going to take a village. This mountain we're going to climb, and this POLG Summit's not going to be done by any one group. It's going to take all of us. And so to hear from more people in this group that are making the way for us, I'd like to invite Luca Bolliger to share with us about the POLG Foundation Toolbox and some of our funded research projects. This will be quite a tour de force, Luca, for you to cover in 10 minutes, but please have at it.
Thank you very much, Brian. Good morning, good afternoon, and good evening to everybody. Very pleased to be here. So I try to share some slides as well. Do you see my screen? Okay,
there it
perfect.
is. Yes, sir.
Perfect. So let's see if it's moving. Okay, perfect. So as you know, the foundation started its activity in 2022, and in 2023, the foundation was already able to support 4 very important projects that are described here. I will try to go very quickly over them. So the first one we supported is Professor Mutha in Boston, and the goal of Professor Mutha's project is to characterize all possible POLG mutations and try to identify a strict link between the gene modified and the point mutations within POLG and its biological effect in cell lines, so that we can have a map about the structure-function relationship within POLG point mutations. The second project, we supported Professor Suomalainen. She's based in Helsinki. And this project is completely different because Professor Suomalainen has access to a Finnish POLG patient cohort. And this is providing directly insights into the human disease because these patients are homozygous for their POLG mutations. And thanks to this cohort, Professor Suomalainen is going to be able to understand better the disease and what externalities are affecting the disease, but also will help in designing possible targeted interventions to provide the therapy for these patients. The third project is supported by Christina Liang, and she's based in Norway, very close to Omar. And in this case, Christina is using cell lines, human cell lines that can be pre-programmed and can be used to create organoids, so small organs that essentially correspond to the human one, but they are much easier to manage and to control. And these small organs in a dish will help us hopefully in designing and creating new molecules that can be used as therapeutics. The fourth project is a project supporting Dr. NG in Newcastle. And in this case, as already mentioned before, the nature of this disease, which is an ultra-rare disease, necessitates to design proper endpoints so that we can properly design drugs and measure the effect of these drugs in this small patient population. This year, and this is new, a press release will go out in the next week, is a collaboration between the POLG Foundation and the Rada Zocco Foundation, a foundation based in Switzerland. And thanks to this support we received from the Zocco Foundation, we are able to support 5 new projects. So the first project is a project supporting Dr. Amy Goldstein and Dr. Mary Kay Koenig, and they will talk about their project afterwards. Here the focus is to create a global crisis response database so that physicians will be able to access key information, even if not experienced in treating POLG patients. The second project is supporting Dr. Sabine Fuchs in the Netherlands, in Utrecht. And in this case, Sabine's goal is to design an in vivo gene editing therapy for POLG patients. Obviously, this initial step will be done in animals, but the goal has already been done at CHOP is to have a POLG in vivo gene editing therapy. The third project we are able to support is supporting Dr. Caroline Su in Sydney, in Australia. And in this case, the group of Caroline detected a strong correlation between POLG and Parkinsonism, and we want to better understand this link, and potentially what we will do with POLG will help also Parkinson patients. The fourth project is supporting Dr. Carlos Moraes in Miami, United States. And in this project, the goal is to be able to engineer mitochondrial DNA in the muscle, so that we can correct some of the mitochondrial mutations that are popping up in the muscle. And the last one is a project that is not per se scientifically, but it is very important because we generated some key information that will help physicians in better identifying and understanding key characteristics of POLG patients. And we also created some videos so that people can really have a sense and an understanding what the research is doing for POLG patients, what the clinicians are doing for POLG patients, and how patients are living with the disease. We generated also some tools that we offer to the industry. One of the first tools that the foundation decided to invest in is to generate a mouse model where these mice are harboring POLG mutations, and these models are going to be very important to understand the disease, but they're also going to be key in order to test potential new therapeutics. And we were able to generate these mice thanks to a collaboration with the Jackson Laboratory, the University of Padua, the Institut Imagine in Paris, and the Lilly Foundation. And needless to say that these models are freely available and can be purchased at the Jackson Laboratory or can be acquired through the University of Padua. The second type of toolbox we invested in is iPSC cell lines. iPSCs are induced pluripotent stem cells, and this is a very useful toolbox because it allows you to take cells from adult patients, reprogram them, and differentiate them in different type of organs, facilitating the understanding the disease and potentially the drug discovery. We were able to pursue this project thanks to a collaboration with the University of Geneva. And if people are interested in accessing these cells, they can contact us and we will organize a shipment of these cell lines. And in the end, in order to foster the collaboration and come up with new ideas and share the tools that the different stakeholders generated, we started having fireside chats where our POLG grantees, share ahead of publication their achievements, their ideas, and we already had 4 of them, and it's really a lively and productive investment for everybody because we already could channel a number of collaborations and a number of interesting idea exchange. So this is all on my side, and if you need more information or if you want to have the slides, please do contact us and we will be able to share those slides with you. Thank you very much.
Thank you, Luca. Quite a bit of research sprinkled around the world, all focused on POLG. Fantastic. Would also want to make sure I recognize Liz from Lilly Foundation. I'm glad you had this slide up there with the partnership that we do with them. And next up, could I please invite Stephanie Harry and Kiraman from MitoAction? They'll each take 5 minutes and tell us about what they're up to. Please.
Thank you so much for having us, Brian. And it's just so exciting to see all of the collaborative efforts and everybody that is on the call. I'm so glad to be here. My name is Stephanie Harry. I'm the patient support coordinator at MitoAction, and I do a lot of different things at MitoAction, but one of the things that I enjoy the most is I help lead our support calls, and we have a Friday support call that anyone can come. It's at noon Eastern Standard Time. But through that process, I've been here a couple years now. One of the things that we realized is that it's really special when you can kind of have these support calls that are really specific to different conditions. So we have an FAOD support call, we have a CPEO and me support call. And so I got connected with Brian a couple of months ago, and we started discussing the idea of having a POLG specific support call. So we had our first one last month, and we actually have one tomorrow at 11 a.m. Eastern Standard Time. And this is a call that I lead with Josie Van London, who has POLG. And these spaces are really important. So if you're on this call and you've never been to a support call, I even hesitate to call it that, just from the standpoint that sometimes that deters people. But this is a collaborative space where we get to share and receive wisdom. And so if you've never had that experience of sitting with other people with POLG that understand POLG, and just sometimes even just being with people that understand the terminology and what it's like to live day in and day out, it's a wonderful space to give and receive support. So I always encourage people, if you're having a good day, great, come. If you're having a bad day, come, because that's how we support each other. So I just encourage anyone, if you have questions, I'm going to put my email in the chat. And this group is, the group becomes like family, and it is about the patients. So again, that's tomorrow at 11, and I'll turn it over to Kyra.
Thanks, Stephanie. And thank you, Brian, and to the POLG team for having us. And we are just truly honored to be able to walk with you guys on this journey and help provide support to this patient community. It's truly an honor. And so I'm excited to share that we are planning in the next 24 hours to submit an application to the FDA to co-host with the POLG Foundation an externally led PFDD meeting. And for those of you who aren't familiar, it's an ELPFDD is the acronym for it. It's an opportunity for us to come in front of the FDA and share the patient journey and the burden of the disease and the unmet needs of the disease. And from your perspective, what would be meaningful in a therapy? So as the FDA has applications that are coming in front of it to consider for approval, they truly understand POLG and the impact that it has on your lives, and making sure that the outcomes for those therapies are the best that we can offer for this community. So there will be opportunities for the entire community to get involved. The meeting will be virtual, and we're looking, our date request will be either at the end of 2026 or in the very first few months of 2027. And we'll be looking for patients and caregivers and family members to join us and to share your story. And it's an incredible opportunity to have the ear of the FDA. The entire FDA is invited to join and listen in, but it's your meeting. It's about the patient voice and being able to share that with the FDA and regulators. We will have leading clinicians and researchers that will talk about the biology of the disease and therapy development and things like that. But the meeting really is about the patients and the families. And so be on the lookout in the course of the next couple months as we find out the meeting has been approved and the date, and we'll do a community survey that will also help guide the content and the things that we need to focus on in that meeting based on what you're telling us is important to you. So there will be opportunities for you to participate leading up to the meeting as we prepare, and also live at the meeting. So we're really excited and honored to work on this effort with the POLG Foundation. We're incredibly honored to represent the patient voice with the FDA and ensure that you always have a seat at the table. So thank you so much, Brian. We appreciate it.
Thank you, Kara and Stephanie. And I, of course, want to make sure I acknowledge that there are plenty of fantastic patient support groups out there. And Margaret Moore from the UMDF has been a champion in this space, and other organizations as well. Just wanted to recognize that Stephanie has decided to pull together a specific POLG group. And I think if those of you are interested, please do. But if you have resources and patient support groups already in place, please, by all means, utilize what's there for you. And I will then, I did want to mention, forgive me, Luca, after your section, I saw Carlos Moraes and Christina Liang on the call here today. So I wanted to say hello to them and thank them for their great work. And Carlos, you're not wearing your POLG hat. No, not today. All right. Well, next up, keeping us on time, I'd like to invite Dr. Amy Goldstein and Mary Kay Koenig to tell us about what they've been working on. Please.
Thanks so much, Brian, and hi, everybody. Thank you so much for inviting us to share what we've been working on. Can you see my slide okay?
Yes.
Okay, great. Okay. So Mary Kay and I are going to present this together, but we've been working very hard on the POLG Crisis Response Team project, and it has really taken off and is gathering more and more momentum as time goes on. So this is a project that we applied for as part of the Superhero Award, and we're very grateful to receive that, so thank you. But this is basically what has been happening and pretty much around the world is when a patient with POLG is having symptoms, is in crisis in the hospital, what has been happening is someone will reach out to one of us and say, what do I do? The patient's having either respiratory problems or seizures, uncontrolled status epilepticus, and we need help. And so what we started thinking is there's a lot of knowledge that we've all accumulated over time, and there's no home for this. There's no database for this. A lot of us talk amongst each other, but there's no publications that summarize all of this. So we really wanted to try to figure out how can we utilize the existing medical knowledge and all of our collective resources so that we can improve the outcomes for POLG. So we want to develop a resource for physicians caring for POLG patients using this collective knowledge and be able to share that. And we want to be able to communicate that to physicians who need it at the time of this crisis. And I'm going to let Mary Kay talk about the next slide.
Yeah, so adding on to what Amy said, this kind of all developed from a project that we started about 2 years ago with the Mitochondrial Medicine Society. One of the things that happens very frequently, and I'm sure all the physicians on this call are aware of this, is just like Amy said, a patient becomes in crisis. And as a physician, we want to do everything we can. We reach out to our colleagues. And one of the things that starts often is these email trains where we start, what would you do? And sometimes you can get 10, 15, 20 physicians on these email trains. What would you do? And we all start answering, well, we did this and this worked and that didn't work. And what would you do now? And 3 days later, we're reaching back out saying, this is the status of the patient now. What should I be doing now? And we're using our collective knowledge worldwide. And so we said, well, we should really try to collect this information that everybody has. And so we started doing this, Amy and I, about 2 years ago as a project with the MMS, and we decided to really hone this, collecting this real world experiences during crisis situations. And so we started kind of collecting these email trains, trying to get all of the information from the physicians that had already been curated, almost the suggestions, recommendations, things people have tried, and trying to follow up on, well, what happened to the patient? Did this work? Did it not work? And so now we're trying to then compile these into a series of things that experts have recommended and create a repository database of what has worked, what's been recommended, not just in the US, but around the world, and develop a repository so that any physician at any time can go and find this information and look at what has been done in the past to try to help a patient and has it worked. And so the next thing to do would then be to try to organize those responses to separate them into different kind of treatment goals. So, what do we do for patients in status epilepticus? What do we do for patients who are respiratory failure, liver failure? I have a patient in the hospital right now who's vomiting. So what do you do in the different situations? And so the goal is to make this information readily available in real time so that any physician from around the world is able to take this collective expert knowledge and see what's been done. If we don't have randomized control trials, at least we can have a repository of all the expert information.
So what we've done so far, as Mary Kay just said, the Mitochondrial Medicine Society, along with the Mitochondrial Care Network, we've already been working on this, and we've been doing this with a lot of surveys. We have some consensus guidelines that have already been published, but at the most recent meeting and getting ready for the recent meeting in June last summer of the UMDF, we sent out this question in advance, and this was a very specific POLG question about what would you do in this clinical scenario. And this was along with other questions that were based on different patients with mitochondrial disease in different states of different clinical situations. And again, physicians were asked for each instance, what would you do now? And what would you do if that didn't work? And then Mary Kay brought this with her clickers to the UMDF meeting live to ask the audience.
Yeah, so then we gathered a group of mitochondrial experts into a room, and we asked them, how many of you would do this? How many of you absolutely would not do this? And how many of you might do this? And so these were all of the different, you can't read it, it's too small, but these were all of the different responses we got about things that people would try. So they would consider transplant, they would call the the hepatology team, they would give N-acetylcysteine at high doses, they would start lactulose, just a whole bunch of different potential treatments that people would have tried. And what we wanted to know was how many people would actually do this, how many people absolutely would not do this. And so I have a medical student who's actually now a child neurology resident who has collected all this stuff. She started organizing all this information. And the goal is, again, to take this not just from the liver failure, but now we're going to take this into status epilepticus. We're going to take this into the various different crisis situations that people will develop. And we're going to try and we're not just going to try, we're going to do it. We're going to expand this out to all the different crisis situations, and we're going to try to get a better understanding of what physicians would do. We're also going to broaden this beyond just the cohort that was at the UMDF. We really want to get a global perspective, not just what people are doing here in the US, but what are people doing worldwide.
Thank you. So right now there's a manuscript in preparation to distribute this information that we gathered at the UMDF meeting regarding current practices in acute management. And we are continuing to field calls and emails through the POLG Foundation from physicians caring for these patients in crisis. And these are some of our future plans. Go ahead, Mary Kay.
I was going to say, so we've developed what is now being called the Crisis Response Team. So Stephanie Harry at MitoAction has been helping us by serving as a liaison for patients and families during crisis situations, helping connect their physicians to appropriate resources. And Brian at the POLG Foundation has been serving as a liaison for clinicians, helping connect them directly to the crisis response physicians, who is Amy and I. And if there's any other physicians out there who would like to join this team to serve as a resource for physicians from around the world who are reaching out, asking for help in a crisis situation, we'd love to have you join our team. And go ahead.
Well, I was just going to say, and the information, we will be making that available online, and we will be making sure people know where to go find that information in addition to the publications, but it will be on websites and links on websites. One of the things that Mary Kay just touched on is that we do want to have a broad reach. And so really, we've just been surveying the mitochondrial community. I was asked to attend this wonderful meeting in April on behalf of the POLG Foundation called Chimes. This was a monogenic epilepsy meeting again in April, held by the University of Chicago, but it brought in many different people that are active in the epilepsy world. And I just had a follow-up meeting at the American Epilepsy Society Society. And this has really allowed me to work very hard on the status epilepticus protocols. I'm now working with the Rare Epilepsy Network, the Pediatric Neurocritical Care Research Group, the Norse Foundation, which stands for New Onset Refractory Status Epilepticus. So all of these groups are now kind of helping to gather what's going to be a very critical resource for us, which is how can we help children and adults in status epilepticus who have POLG. And then we're going to turn that into a consensus protocol so that we can take the best course of action for status and EPC for our patients. And so we both want to thank you very much for the Superhero Award, and we are going to make very good use of this time and the award. Thank you so much.
Thank you.
Fantastic, Mary Kay and Amy. Awesome. For those, and many of you know, families out there, you know that in rare disease space, you know, it just, every case by itself stands alone. And how do we learn from that without someone pulling it together? And Mary Kay and Amy, I can't thank you enough for having this idea. And the POLG Foundation is 100% with you and looking forward to seeing where this evolves to improve the care for POLG individuals. And that's not without other clinicians out there. I see Shamima Rahman, I see Bruce Cohen, I see Michelangelo Mancuso, of course, with Amel and Omar. And I know we even have Dr. Anina Enderle from Switzerland on this call. So for the physicians that are on this call, please connect with Mary Kay and Amy, and let's build this up to be a robust database. Okay, thank you so much. If I could please invite Dr. Robert Picathley and his team from UCL to tell us a bit about LifeArc and some of the exciting gene therapy work that is underway, hopefully sooner than later. But Robert, please.
Hi, Brian. Hi, everyone. So hopefully you can see our slides. So I'd like to just start by thanking you, Brian, and the POLG Foundation for inviting us to talk about the work that we're involved with developing dual AAV delivery systems of POLG. You can go to the next slide, please, Jana. So this is work that's funded by LifeArc and also Muscular Dystrophy UK and heavily supported by the Lilly Foundation in the UK. So LifeArc is a not-for-profit self-funding life science organization which focuses on rare diseases, and it specifically invests in projects aimed at translating discovery science into clinical application. And it's funded several rare disease centers focused on this mission. And so the LifeArc Center for Rare Mitochondrial Disorders is a platform that connects specialists, patients, and researchers across the UK, primarily UCL, Cambridge, and Newcastle, with Patrick Chinnery being the lead for the centre in Cambridge. So it was launched at the House of Lords in May, and it has a number of workflows, cross-cutting workflows that include diagnostics, improving diagnostics, biomarkers and outcome measures, with a particular focus on MRI as an outcome measure in mitochondrial diseases, and also the development of small molecules and gene and genetic therapies. And the work that we want to just share with you today is on POLG, which is one of those particular projects. So Jana Aref is a PhD student in my lab who's going to speak to this, and she's doing that on behalf of the whole team that's involved in this, along with Ruben Shi, who's also heavily involved in these approaches in the laboratory. So I'll hand over to Jana.
So good evening, everyone. I'm really excited to be here and to tell you a bit more about our gene therapy approach for POLG. So our approach will be a gene replacement approach. So the idea is that you have the mutant dysfunctional POLG gene. So the goal is to replace it with the correct gene, which will then be translated to a functional POLG protein. So one of the big challenges for gene replacement for POLG is that the POLG gene is quite large. So it exceeds the AAV cargo capacity or the packaging capacity to deliver genes. So to address this issue, we'll be doing two split gene strategies. So the first strategy would be the Stitch R strategy, which will be led by Rubing. And this will be splitting POLG on an mRNA level. And then I will be doing the split intine strategy, which will be splitting POLG on a protein level. So just an overview of the Stitch R strategy. So this strategy will rely on ribozymes, which are naturally occurring genetic elements. They're RNA sequences that can be split and they are able to self-cleave. So the idea is that you split the POLG gene into two halves. You attach the ribosomes and then you package them into AAV vectors. And once they are delivered, the ribozymes will facilitate the fusing of the two POLG gene halves, and it will self-cleave, and it's a scoreless process that will leave you with the correct POLG protein. And this strategy has been used successfully for muscular dystrophies, so it proved efficacy in dysferlinopathy as well as Duchenne muscular dystrophy. So for the split intine strategy, it's very similar. So, but it relies instead on split inteins, and split inteins as well are genetic elements that facilitate the splicing or the joining of two smaller protein fragments, and then they self-excise as well. So this would allow us to deliver the large genes such as POLG by splitting POLG in half. Then you attach the inteins. Again, you package the two halves separately, and you deliver the gene therapy. And once they're translated into a protein, the inteins will recognize each other. They will allow the two POLG halves to join into one functional protein and will self-excise out. And again, this has been used successfully to treat Duchenne muscular dystrophy. So we're very hopeful that either one of these strategies or both of them will be successful for POLG as well. So just an overview of the experimental plan. It's very similar for both projects. So the first stage would be to validate the strategy and validate the vector design. And once you have multiple candidates, we'll move on to in vitro testing or testing in cells. And this will be done in fibroblasts taken from POLG patients, as well as cells taken from POLG mutated mice. And then finally, the third stage, once we have optimized the candidate, will be in vivo testing or animal testing, which will be done in POLG mutated mice. And I just want to stress the importance of this third stage. So this is done in collaboration with Carlo Viscomi at the University of Padua. And we're very fortunate that Carlo has already generated multiple mouse models for common POLG mutations, and he's already characterized them. So this will be a great advantage for this project and will hopefully maximize the chances of success for these therapies and help us reach the point where we can use it on patients. So I'd like to just thank everyone involved specifically in this project, as well as everyone involved in the LifeArc initiative, and I'm happy to answer any questions. Thank you.
Wow. Jana, thank you so much for that. Robert, thank you for that. If you can't get a sense, there's a lot of passion and brilliance and innovators here. I am just awed. But we're not done. There's more still to come. So I got to stay on time. If there are questions, please put them in the chat and we'll make sure Robert and Jana have a chance to take a look and see if they can respond to them. And if there's time, we'll try to tackle them. I want to take a moment and thank some individuals that have really been crucial of fueling our mission. And when I say fueling our mission, you know, we do one thing. We raise awareness and money to be able to push into research. And the research is to get us to the clinic and to get us to medicines for POLG. And I would like to acknowledge some people that are on this call and some that aren't on the call. But I wanted to thank Marius, Corinne, Oxolin, Karen Holland and her family, Frederick's brother and sister are on this call. And Mansour Shikarshi ran a marathon and raised a ton of money. And I want to say hello to Mansour and Charlotte and Frederick's brother Alex and his partner Hedda, if they're on the call still. We have Mark Cunningham, an uncle on the call, and just great to have family supporting this. And I would like to also ask Harrison Dodds and Ellie, please, if you can introduce yourself and if you want to share a little bit about your story and your family, and then we'll open up the industry partners, and we'll start with Stealth and then Pretzel, and then UCB if UCB wants to say anything. Harry, do you think you can pop up on the screen?
Sure.
Please.
Good morning, good afternoon, good evening, wherever we are in the world. And Brian, POLG Foundation, thank you so much for putting on today's event. Well, my name's Harrison, this is Ellie, my wife, and we were first affected by POLG in July of this year when we found out that our young son, Charlie, 1 year old, was diagnosed with POLG. I suppose from it, it's no news that what any parent wants to hear, and we instantly were sort of plunged into a state of shock and despair, and we're still sort of working our way through that as we try and progress and find hope and find a potential cure for Charlie. So, as I said when I first came on, I just want to thank you very much, Brian and the POLG Foundation, for putting on such an initiative and enabling a community to come together and provide us parents who are facing it on a daily basis, provide us the much needed hope that we look for on a daily basis, if you like. So thank you.
Thank you, Harry. Ellie, great to see you. Thanks for joining us.
Thank you. Thank you for having us.
All right, if I could invite Katie Landis from Stealth, if you would, please, Katie.
Thanks, Brian. I'm going to go ahead and share my screen. Is everyone able to see my slides?
Yes.
Perfect.
Popping them. I don't know if you want to. There we go. Perfect. Projector mode.
Well, thank you so much for the invitation to provide an update on behalf of Stealth Biotherapeutics. My name is Katie Landis. I head up patient advocacy and government affairs, so not too many things for Stealth. And I am very honored to be here today. Brian, Luca, Julie, what an incredible effort to bring together so many partners. And I have to say, Harrison and Ellie, thank you for your vulnerability, because that's why we do what we do. So I'm going to provide a little bit of an update on Stealth Biotherapeutics, because you all might not be familiar with us. And then I'll provide an update on New Power, which was our phase 3 clinical trial in primary mitochondrial myopathy. So just a little bit of background on Stealth for those who aren't familiar with us. Our focus as a company is really on pioneering mitochondrial targeted therapies, and in particular focus on mitochondrial structure and function. We have clinical experience in a compound called elamiprotide, which is a peptide that is administered by subcutaneous injection. That clinical experience ranges across a number of different conditions, including Barth syndrome, which is an ultra-rare mitochondrial disease that impacts around 300 patients worldwide. We were granted accelerated approval for elamiprotide in September of this year by the US FDA. We also have extensive clinical experience in primary mitochondrial myopathy, including multiple phase 3 trials. I'll discuss that in a little bit more detail on the next slide. Ultimately, as a company, patients and families inform all of our work, so I'm really proud to be joining you today and to learn from you. I think we've learned firsthand throughout all of our development experiences how impactful patient stories are, perspectives, and especially in an ultra-rare disease like POLG. And we hope that those perspectives, many of which I'm hearing today, can continue to inform our work. So just to provide a little bit of background on our new POWER clinical trial, I think folks have been asking for an update on this, and so happy to share a little bit of an overview. First, I'd like to thank all the trial participants, families, clinicians, researchers, and partners like the POLG Foundation. Participating in a clinical trial is not an easy decision to make, so we really appreciate the support of the new POWER clinical trial. Every trial is ultimately an opportunity to learn, and our clinical experience in primary mitochondrial myopathy has involved a lot of education and lessons learned. So for the new POWER phase 3 trial, we ultimately enrolled patients with nuclear DNA mutations causing primary mitochondrial myopathy. We've to date conducted a top-line data analysis, which shows that the trial did not meet its primary endpoint. However, we did observe signals in the new POWER trial, as well as in our previous phase 3 trial, the POWER 3 trial, that are encouraging, particularly in patients with POLG1 mutations. That signal persisted across multiple endpoints. Ultimately, the signals in the POLG1 mutations across 2 phase 3 trials may provide a regulatory pathway forward. Where we sit today is we don't know what next steps look like for 2 reasons. 1, we have to complete the data review, and then 2, we have to meet with regulators at the US FDA. Our goal is to meet with regulators in the first half of next year to understand the potential regulatory path forward with the data that we've already amassed to date. So I hope to be able to come back to you next year and share some more updates. I think one other point I wanted to touch on was our expanded access program, as Brian flagged this for me. I just wanted to reiterate Stealth's expanded access program policy for those who may be curious. Our goal is to ensure continuity of care for all mitochondrial patients currently receiving elamiprotide through the expanded access program, who, in the opinion of their treating physician, are demonstrating clinical benefit. Any changes to the program will always be communicated through a treating HCP. We are still reviewing requests for expanded access on a case-by-case basis for those who require emergency access. At this time, we're not reviewing requests for non-emergency access, and the EAP program is always subject to change. So I just wanted to reemphasize that if you are currently receiving therapy through expanded access, we are committed to, ensuring continuity of care for the program. I'll stop sharing my screen there and just say thank you again. This has been an incredible effort to see so many faces on one call to talk about POLG. It's definitely something I'm going to bring back inside the company to keep everyone working hard and hopefully having some conversations with regulators soon. I will pop my email into the chat if anyone has any questions or would like to connect. And thanks, Brian, so much for the time.
Thank you, Katie. And I think I saw David Brown also on the call. I definitely want to thank David for joining us as part of the Stealth team. Okay, wonderful. I'd like to invite Julie Jones from Pretzel. Julie, if you could, take us away.
Let me share. Okay, can you guys see that?
Yep, we can see it.
Okay, great. Well, thank you so much, Brian, and everybody for coming to commit together. This is such an important topic for listening to the patients, listening to all the activities from the patient foundations, and then it's great to hear from the companies. So if you haven't heard of Pretzel Therapeutics, we're a small biotech company. We're based in the US and in Sweden. Our core mission, sorry, it's a little bit slow, is creating a new paradigm in the bioenergetics space through transformative mitochondrial DNA approaches to treat patients with high unmet medical need. Our flagship program, and what I would like to talk to you guys today about, is PX578. So PX578 is a small molecule brain-penetrating POLG activator, and we are currently in phase 1. We're really excited about this program. I'm going to show you just a little bit of a non-clinical data. We haven't really been sharing this to this point, so you guys are some of the first to see this. So we have substantial preclinical efficacy in multiple in vitro and in vivo models of MDDS, or mitochondrial DNA depletion syndrome. We've seen that PX578 in the non-clinical space can increase mitochondrial DNA, restore OXPHOS. We have shown efficacy in 2 different MDDS mouse models. And what's really important we see for the POLG patient is that this compound is, again, CNS penetrant. So we are focused on not only the brain, but the rest of the body as well. So just briefly, we look at how PX578 is working both, again, in cells and animals. The graph here on the left is just 3 examples of our biochemical assay looking at how PX578 can increase mitochondrial DNA, and that's depicted here in the response. And here I'm showing you 3 of the most common POLG mutations. We've tested PX578 with over 50 mutations and know it's a biochemical potency to a number of different POLG mutations. In addition to biochemistry, we look at patient-derived cells. So we bring cells from a number of different patients carrying POLG mutations and look at the response in vitro. And so in a normal fibroblast, you have a very low level of mitochondrial DNA. And just within a few days of incubation with 578, we see a really great restoration of mitochondrial DNA. And in our cells, it's not just about mitochondrial DNA, it's about how the cell is producing energy. And so with that, we look at both ATP production and OCR, which is oxygen consumption rate. And we can see within our fibroblasts, we see a really significant increase in OCR. And also in more energetically demanding cells, such as neural stem cells carrying POLG mutations, we see a really great restoration of OCR and an increase in mitochondrial DNA. So I'm going to show you next some really exciting data from one of our animal models. We're really excited that the POLG Foundation supported another POLG mouse model that we're hoping to work with very soon. But for now, I'm going to show you some data on our TK2 mouse. And so this is a mouse of MDDS, so they have severe mitochondrial DNA depletion, which leads to an increase or decrease in survival. So this is depicted in the black bar. So we start treating these mice on day 4, and as you can see, we get a significant increase in survival of these mice. This is very similar to what the nucleoside therapy, which UCP, which just got approved, which is very exciting for the community. In addition to this increase in survival, we see a significant increase in copy number of mitochondrial DNA. So I can continue more about all the exciting in vitro and in vivo data we have, but what's really exciting for PX578 is that we are in the clinic right now. In early 2025, we started our phase 1 study in healthy volunteers in New Zealand, and we're continuing that work right now and targeting to complete that by the end of, or the beginning of next year. Following that, we are on track to start our first inpatient study in POLG in the second half of 2026 and aiming at seeing proof of concept data by mid to the end of 2027. So that's all I'm going to talk about today. There's a lot more exciting stuff coming out of Pretzel, and I'd like to thank you for listening.
Thank you, Julie.
Yeah.
Thank you so much. Would love to say hello to Boyd Brooks at UCB if he just wants to say hi. If not, we're just so grateful that you're here and you have great interest in POLG and the mito space. Boyd, I don't know if you're on the call still or not.
Yeah, hi Brian. It's Brooks. Yeah, sorry, I have
Brooks.
two
There you go.
first names, so it's
Brooks,
it's
boy.
a liability.
Apologies.
No
There you
worries.
go.
It's great to be invited here today. We're really excited to have the opportunity to learn more about POLG disease. As was just mentioned, Kygevvi was recently approved by the FDA for the treatment of TK2 deficiency, which is a related mitochondrial disease. We're working hard to get approval, we hope very soon, in Europe. And then obviously thinking about, potential opportunities outside of that disease. Again, very pleased to be here, and it's really exciting to see all the energy and the large audience who are also working on POLG disease and really trying to make a difference for these patients. So thanks a lot.
Fantastic. Thank you, Brooks. Wonderful that you're here. I'm sure it doesn't escape everyone on this call that we have multiple different stakeholders from various different sectors, and we're all together because we're all on this team to solve POLG. And there's no other way it's going to happen. So I hope that we can say that we're coming into the end of this year feeling optimistic. And I'm very cautious that I don't want to hype anything. Nothing you'll ever hear Brian want to hype. But I want everyone to feel that there is a sense of hope, that there's some damn smart people working hard on this, and we got to solve it together. So if the POLG Foundation serves as the vehicle to help pull people together and make this happen sooner, then that's our role, and we're thrilled to do it. I had a couple other people I wanted to acknowledge on this call. Jen Adams is one of our board member. Jen, I don't know if you want to say hello if you're still on the call.
I just want to thank everybody. I'm sitting here in awe and really just very deeply thankful and excited by everything. Just really, and Brian, thank you, because this is extraordinary. And it's ending the year on a wonderful feeling of hope.
Wonderful. And we have a new team member with the POLG Foundation, Mindy Leffler. I don't know if you want to just wave, say hi. I skipped over you earlier. I'm sorry. I don't know if she's able to do so. I know she's traveling, might be in an airport somewhere.
Right, I'm just on a plane, so it should be quiet.
Okay, great. I just want everyone to know who you are, and you're critical to our pioneer work. And I also see Matt and Catherine, who have been part of helping us with some legal perspectives. And I want to thank you. I won't make you say anything, Catherine or Matt, but thank you again for just stepping into our world, of which you see there's a vibrant community of some amazing individuals. And I'm going to pause here and see if anyone has burning questions they want to ask out loud. There are many in the chat, so I hope questions that were directed to the speakers, if the speakers can please make sure they can try to tackle them on the chat side. And then again, I want to remind everyone at 2:30 to 3, Stephanie's willing to stay on to meet and debrief with anyone from the family patient support side. So let me see how we're looking from the big screen of lots of people. Anyone have a question you'd like to ask? Just raise your hand.
Okay.
Hi, Brian. Harrison here.
Please.
Can you hear me okay?
Yeah, please.
Yeah, okay. Just a question for Stealth and Pretzel. Just, I suppose, given the time critical nature of the disease and how crucial it is to ensure patients are seen to as quick as possible, is compassionate exemption something that would be considered for pediatrics on the up and coming POLG trials, in particular the Pretzel trial that's heading to North Europe in late 2026?
Katie, did you want to go first?
I think it was more directed at you, but I'm also happy to answer.
Yeah, so at this time, we're not ready to provide that type of guidance of what the company strategy will be. The PX578 is, again, a novel small molecule. And so we do need to be able to learn its PK, safety, tolerability in patients before we go into really severe patients. But it is something that we continue to discuss that strategy. So.
And then
Katie,
from our side, as I said, we're still evaluating regulatory next steps. So whether that means the data we have in hand is a path forward versus if we have to run another trial. But we have extended expanded access experience with elamiprotide across a number of mitochondrial conditions, and we continue to review cases on a case-by-case basis. So I will just put we have a page on our website, and I'll just put the email inbox there. All of that would involve a treating HCP making a decision. So would just encourage you to look at that part of our website for more information.
Okay, thank you. Given your experience in working with pediatrics, would there be any potential synergy between Stealth and Pretzel going forward?
I think our
Sorry,
mechanisms of action are different.
Yeah.
So,
yeah, I
but
appreciate I'm speaking from a layman's perspective, but just I just thought I'd ask the question. Yeah.
no, I think it's a fantastic question. Different mechanisms of action, which is exciting, right? So I think that's part of probably the goal of today's conversation is to show that there are different potential ways to potentially treat POLG 1.
Yeah.
But we learn from each other all along the way, trial design, etc. So I don't think synergies is the right word, but certainly
Or collaboration. Yeah.
mutual learnings.
Okay, understood. Thank you.
And what UCB has, I think, done in their recent approval, and Barth too, but for the community, it's wonderful. We have 2 new approvals right within Mito, and that just helps fuel industry to get involved more, invest more. So we're thrilled when both Barth and TK2 patients received their recent approval. It helps us all.
So Harrison, one thing that I wanted to say that is true from your question is that drugs with 2 separate modes of action may actually be synergistic when used together. So even though the trials have to be done separately initially, eventually the clinical impact of this might be best felt when the 2 come together, and are used together. So those are further hope for studies down the line.
For sure. Thank you, Robert. Thank you.
Thank you, Bob. Thank you, Harry. Great question, Harry. The idea that could there be a need for a cocktail to really hit the different facets of the POLG disease. As we know, it manifests in different organs, and the blood-brain barrier has a different compartment. And so it's not beyond my thinking as Bob's and many of you on this call that it may take more than one agent to really solve POLG. Thank you for that. Julie, I think there's one other question that just popped up on the chat. I've not seen all the chat questions, but
Yes,
I
I was
see one
trying to
that's
figure out how to
for you.
respond.
Okay,
No,
okay,
so
if you can.
yeah, Pretzel, we are a global company and we are focused on a global strategy. So I can't say specifically what countries because we're working through that right now with KOLs. But we do look at this as a global community and a global strategy to reach patients.
Excellent. Anyone else want to raise their hand and ask a question to someone in the group? Okay, well, I want to thank each and every one of you, and particularly the speakers who shared today. And for those that haven't been asked to speak today, oh, Bob, nice hat. We all know Frederick designed that hat. Awesome. Just so everyone knows that this was the first summit that we didn't know if anyone would show. We're delighted to see that people are, and it's very likely there will be more summits to come. And for many of you on this audience, don't be surprised if you get asked to speak at the next summit or thereafter. And so I wish you all happy holidays. And for those that will stay on with Stephanie, please do. But everyone else, wish you happy holidays and thanks for being part of our journey, our crusade, and let's solve POLG together. Take care.